Rakshit Galwa, a grade 12 student from Leo Hayes High School. Climate change has been causing sea levels to rise in many parts of the world, and this will likely continue happening in the future, and climate change-induced seawater inundation of terrestrial environments can be a big problem for agriculture. In this project, I examined Sinorhizobium meliloti and how it responds to increasing levels of artificial seawater, ASW, in its growth media and attempted to identify genes required for the survival of bacteria in a hypersaline environment. Meliloti represents an agriculturally and ecologically important group of nitrogen-fixing soil bacteria that promotes the growth of legume plants by forming a symbiotic relationship with them. In this project, there were three main focuses: how meliloti survives in artificial seawater in the short term, how it persists in artificial seawater in the long term, and what genes behind its level of resistance to the seawater stressor. To start off, I made a solution to mimic seawater at 2 times the concentration and then used it with 2 times LB broth or agar to create the desired concentrations of ASW that were used throughout the project. For the short-term experiments, I streaked colonies of pure meliloti onto solid LB agar of increasing ASW concentrations and inoculated the test tubes filled with LB broth and increasing concentrations of ASW. I found that the bacteria did not grow at all once 0.75 times ASW was reached or exceeded on solid media, and in liquid media, the optical density and thus growth of the 1 times ASW was only slightly more than the test tube with no inoculation. This demonstrated that meliloti does not grow well in high seawater conditions. To replicate what it would be like in nature, we put topsoil in 9 series of test tubes, each for data collection days of 2 weeks apart, and then inoculated them with cultures of meliloti and increasing ASW concentrations. 3 days after 100 microliters of each tube from a series was spread plated and inoculated, the number of colonies was assayed and the number of colony forming units per milliliter were calculated. This was done until the COVID-19 lockdown inhibited the collection of the last few data points, but the data that we do have does show that meliloti survives in 0.5 times ASW, similar to what it would if there was none, but in 1 times, it dies off very quickly. While the long-term experiment was going on, we were trying to replicate a mini TN5 transposon library created by PhD student Yukin Benzig onto 1 times LB agar and then onto a sublethal ASW concentration. The colonies that don't grow on the ASW plate have the transposon insertion in a gene required for growth in or on seawater. We obtained 1 isolate, which we then named NB016, with impaired growth, which underwent inverse PCR and was sent to Genome Quebec for DNA sequencing. We identified the gene where the transposon inserted itself in as SMC03845, and it did so between nucleotides 89 and 90, and this gene was of an unknown function. However, looking at the amino acid sequence, it contains a CSSC motif, one that indicates the resulting protein is redox active, meaning that it may help the bacterium survive in oxidizing environments. Since there's often a link between survival in oxidizing environments and high salinity, it is very likely that the gene directly affects the survival of bacterium in seawater. From my results, we can see that meliloti cells will not grow well in high salt conditions, and in soil saturated with seawater, they die much more quickly than in soil without seawater. The gene that we identified, SMC03845, in mutant meliloti, which were considerably less tolerant to our artificial seawater, is of an unknown function. And this is actually the first report of a phenotype attributed to that gene. Future studies will be aimed at understanding the function of the gene's proteins, and our work identifying this gene's function will pave the way to enable engineering of agriculturally important bacteria to resist high salt concentrations in soil, because events like climate change will cause sea levels to rise. I am passionate about agriculture since I'm from a farming background, and I wanted to do research on a topic that has the potential to improve the quality of crops all farmers can grow. Every learning opportunity in biology is very enjoyable, and I wanted to do something that involved the growth kinetics of meliloti to delve deeper into the bacteria's growth on ASW, but the final data was just too complex and impractical to continue, and sometimes that's okay. Before I wrap up though, I would like to thank Dr. Sean McClellan for being my mentor and for helping me grow and learn throughout this entire process, And I would like to thank everyone who has made the Sanofi Biogenius competition possible this year. Thank you.